students, in this session we are going to see some of the important DNA viruses. So we have learned the mnemonic for DNA viruses, and you know some of the DNA viruses by now. So we'll see the classification of DNA viruses, how we can classify. We have not learned any proper classification. We have just learned what are the DNA viruses. So we can just see whether you're finding out all the important DNA viruses here. The 2H, herpes viridae, hepatnaviridae, A, adenoviridae, P for papilloma, that is actually pepovaviridae, including papilloma and polyoma, pox viridae and parvoviridae. All of them are here. And how they have classified this is, you can classify them as double-stranded DNA and single-stranded DNA viruses. We know there is only one single-stranded DNA virus, that is the smallest DNA virus called the parvovirus. Actually, parvovirus causes a disease called fifth disease. It occurs in small children, leading to rashes over the face. So it's called fifth disease, and sometimes it can cause gastroenteritis also. So that comes under this part, the single-stranded DNA, and parvovirus is non-enveloped. So parvo is a very small virus. It doesn't have any envelope, and it is a single-stranded DNA virus. Now, looking on to the double-stranded DNA viruses, how we can classify them? We can classify them as enveloped and non-enveloped DNA viruses. So the non-enveloped DNA viruses are adenovirus and papillomaviridae. So adeno and papilloma are non-enveloped. So your mnemonic is HHAPPPI, happy. So in this A and P, adeno and pepovaviridae or papilloma virus, both of them are non-enveloped. They doesn't have an envelope. And your parvovirus is also non-enveloped. That is easy to remember because parvo is very small. Otherwise, all others are enveloped, including pox, herpes, and hepatnaviridae. So, this is how you can classify DNA virus into double-stranded DNA and single-stranded DNA. Double-stranded DNA, what are the enveloped and non-enveloped? Non-enveloped viruses are the adeno and papillomaviridae. And enveloped viruses are all the rest, herpes, hepatna, and also pox virus. Pox is a very big virus, so it is definitely enveloped. That is how you can remember the various DNA viruses. So suppose the question comes as classify DNA viruses, you can classify it like that with examples. So we are seeing the most important one. One of the DNA virus we have already learned, that is hepatitis B virus. Hepatitis B virus is an important DNA virus which was dealt along with the hepatitis viruses. So now we are going to see the next important DNA virus, that is herpes virus group. The family is called as herpes viridae. So all the family names end with viridae. And they are subdivided into three subfamilies, including alpha, beta, and gamma. Alpha herpes viridae, beta herpes viridae, and gamma herpes viridae. And the very important feature of this family, herpes virus, is they have the ability to remain latent in the nerve ganglia. Most of the time it is the nerve ganglia. It can also remain latent in the lymph nodes and also sometimes in the glands, depending upon the type of virus. So they can remain latent in the places where they like. It can be a neural ganglia, or it can be the glands, or it can be lymph nodes. So wherever they remain latent. Once you get an infection, you are getting subjected to lifelong latency by the virus. The virus is very difficult to eradicate. So what happens is, at any point of time, you can get a reactivation of the virus. That is the problem with this group of virus. So when you hear about herpes virus, please remember it has the risk of latency and reactivation. That is the important property of herpes virus. Now the size of herpes virus is like 100 to 200 nanometers in size. And you might be remembering the size of a pox virus, which was 300 nanometers. So this one is also slightly bigger one, 100 to 200. And this is having a icosahedral capsid. We have seen what is icosahedral capsid. And you can see it has got a linear double-stranded DNA genome. So it is a DNA, which is a double-stranded DNA, and it is a linear strand like this. And surrounding this DNA, you have the capsid layer, that is called the nucleocapsid together. Outside the capsid, between the capsid and the envelope, there is a layer called as the tegument. Tegument layer is between the capsid and the envelope. And this has got a lipid envelope layer, which is the outermost layer, which is having various protein spikes from that is called the peplomas. So this is the structure of herpes virus. in general in most of the herpes species. I mean, this is the structure of the herpes viridae family. So we'll see how we classify herpes viruses. So this can be a probable question. We have seen there are three subfamilies for herpes viridae family. The first one is alpha herpes viridae, which includes three types of human herpes virus: HHV1, HHV2, and HHV3. You abbreviate this as HHV, human herpes virus. So these are the viruses which are coming under alpha herpes viridae, and you know HHV1 includes herpes simplex virus 1, HHV2 includes herpes simplex virus 2, and HHV3 includes varicella zoster virus. So these three viruses, these are very important viruses for you, that is HSV1, HSV2, and varicella zoster virus, which causes chickenpox. All these three come under alpha herpes viridae. Now, beta herpes viridae, that is the second subfamily. The first one was alpha, second one is beta. Here you have 5, 6, 7. So after 1, 2, 3, you are going to see 5, 6, 7 in beta. And 5, 6, 7, HHV5 is commonly called as cytomegalovirus. There is no specific name for 6 and 7. 6 and 7 usually causes fever with certain kind of rashes in children. You don't have any specific name for them, but HHV5 is the cytomegalovirus, which comes under the beta herpes viridae family. The third group is gamma herpes viridae, which includes 4 and 8. So alpha included HHV1, 2, and 3. Beta includes HHV 5, 6, and 7. And here you are seeing the gamma herpes viridae, which has HHV 4 and 8. So HHV 1 is your herpes simplex virus 1, 2 is HSV 2, and 3 is the varicella zoster virus, and 5 is commonly called as cytomegalovirus. No specific names for 6 and 7. So here this 4 is coming under gamma herpes viridae, and 4 is the Epstein-Barr virus, and 8 is the Kaposi sarcoma associated virus. So this is how we classify herpes viridae family, and please remember Epstein-Barr virus is HHV4 and Kaposi sarcoma associated virus is HHV8. These are also probable areas where you can get MCQs. So this is how we classify herpes virus group. Now we are going to see the different viruses in separate sections. So the first one that we are going to see is herpes simplex virus, which belongs to the alpha subfamily. And what are the members of alpha subfamily? HHV1, HHV2, and 3. So we are going to see HHV1 and HHV2, which includes herpes simplex virus 1 and herpes simplex virus 2. So man is the only natural host for herpes simplex virus. There are two types of HSV, HSV1 and 2. That's what we said now. HSV1 is usually associated with lesions in the upper part of the body. You can remember like that. HSV1 is mostly concentrating on the upper part of body, so it can cause skin lesions on the upper part, oral lesions, and ocular lesions. It can also cause encephalitis affecting the brain. And HSV2 is responsible for majority of the genital infections caused by herpes simplex virus. So the mode of spread of herpes viruses, herpes simplex virus, is mostly by direct contact. The secretions like the saliva will be highly rich in the virus. The virus will be shed in the genital secretions, in the saliva, in the conjunctival secretions. So if you have a contact with these secretions through any mode, you have the risk of getting the disease. So what are the common infections caused by HSV 1 and 2? This is important. This is a question which was asked previously. The question was like that: lesions caused by HSV. So you have to write both HSV1 and HSV2. HSV1 causes various lesions, like see what it causes in the oral cavity. It can cause acute gingivostomatitis, inflammation of the gingiva and the oral mucosa. It can lead to herpes labialis. Herpes labialis, there will be painful blisters in the corner of the mouth and lips. That is called as herpes labialis. And there is a condition called in herpes labialis, it can be like recurrent herpes labialis. If a person has a herpes infection for the first time, the problem is the virus will be remaining latent in the nerve ganglia, in the dorsal root ganglia. Second time, then when the patient gets any other fever, any other kind of viral fever or any other fever, there can be reactivation of these viruses in the ganglia, and they will be going back to the region of the nerve supplied by the ganglia, and they will produce lesions there. So you will get this recurrent herpes labialis like lesions in most people who have had a history of herpes for a once in their lifetime. They get the recurrent lesions whenever they have any other kind of fever. You call that as fever blisters. So you call that as fever blisters. So this is a reason. This is because of the latency and reactivation of herpes virus. Then the other condition caused by HSV1 is keratoconjunctivitis, cornea and conjunctival inflammation, eczema herpeticum in those people who are having skin lesions like eczema, especially children. They can have an extensive herpes lesions along the skin. So it's called eczema herpeticum. Then encephalitis, another condition caused by HSV1. And HSV2 is mostly known to cause the genital herpes. It will be painful vesicular ulcers seen over the external genitalia. Similarly, this is known to cause neonatal herpes because a child during delivery, the baby can get exposed to the herpes virus if mother is infected with genital herpes. So they can have this herpes lesions and other congenital problems that is called as neonatal herpes. And so that herpes can also be transmitted transplacentally, not just perinatally. So transplacental transmission can lead to congenital anomalies in the fetus. Other than that, HSV2 can also cause aseptic meningitis. So mostly encephalitis is associated with HSV1, whereas HSV2 can also cause meningitis. So basically, HSV2 is mostly causing genital herpes and neonatal herpes, whereas the other conditions like the oral lesions, the mucosal lesions of the oral cavity, lips, mouth, and keratoconjunctivitis, encephalitis, everything is caused by HSV1. So this is a question. So please be familiar with the lesions caused by HSV. So how will you diagnose an HSV infection? You have to take the samples. So what kind of samples you can take? If the patient is having these vesicular lesions, you can take the vesicle fluid. That is the best sample that you can take. Or you can take scrapings from the base of the ulcer. So you can take scrapings from the base of the ulcer. That will be very good if you take the scrapings from base of an ulcer produced by HSV. You can take a skin swab, or you can go for a salivary sample, corneal scrapings in case of keratoconjunctivitis, and CSF in case of meningitis. So what you're going to do with the sample, basically herpes is having a special property. It can cause fusion of the infected cells, leading to multinucleated giant cell formation. So these multinucleated giant cells can be visualized by the light microscopy. So though the virus is very small, you need an electron microscopy to visualize the virus. But since this virus is producing this multinucleated giant cells, and also they produces inclusion bodies, you can visualize that with the help of a light microscopy. So you can do a Giemsa stain or a toluidine blue stain for the smear, and the smear is prepared from the scrapings from base of the vesicles or the ulcer, and this smear is stained with toluidine blue. And when you examine the smear, it will be looking like this. You can see this is a large cell with multinucleated large cell faceted nuclei. So you have a faceted nuclei, multinucleated giant cell, you can see here. So these cells are called the Sang cell, and the smear is called Sang smear. So Sang smear is a 2-mark question which can be asked for exam. So what is Sang smear? It is a smear prepared from the base of from the scrapings obtained from base of vesicle, which is stained with toluidine blue. And what will you see in that? You will see multinucleated giant cells with faceted nuclei like this. It will be faceted nuclei, and you will have a ground glass chromatin. So that is what is called a Sang cell and Sang smear. And remember, Sang smear or Sang cells are positive not only in herpes simplex but also in varicella zoster virus, that is which belongs to the alpha herpes virinae subfamily. And I told you it has property of producing inclusion body. What kind of inclusion body? Cowdery type A. So how we have remembered Cowdery type A? This is how we remembered Cowdery type A. A is the type A, H for herpes, and Y for yellow fever. So the two viruses which produces Cowdery type A inclusion is herpes and yellow fever, and they are intranuclear inclusion bodies. So this can also be visualized by Giemsa stained smear, and you can use a light microscope for this. So generally, these are the rare instances where we can depend on a light microscopy for visualizing the virus producing structures. Now, what can you do with the sample other than microscopy? You can do a culture, you can isolate the virus from tissue culture. So, what kind of culture we can use? You can use a human fibroblast cell line, which is a diploid cell line, or you can use HEP2 cells and Vero cells, which are continuous cell lines, or you can inoculate it in the chorioallantoic membrane, and that can also be used for the virus isolation. And how will you confirm this virus has grown in the cell culture? You can use an immunofluorescence staining. That is the best way of demonstrating the viral growth in the cell culture. Otherwise, if you remember, herpes virus is a cytopathic virus. What kind of cytopathic change it produces? Herpes will produce crenation of the cells. It will cause crenation and degradation of the cells. So that can be visualized. If the virus grows in the cells, you can visualize the rapid crenation and degeneration produced by herpes virus. So cytopathic change can also be visualized if the virus grows in the cell culture. Now you have serological test available for HSV, and better than that, if the case is that of a encephalitis or meningitis, you want to get a rapid diagnosis. You can't wait for culture. Never, culture is never a diagnostic modality which is easy. It takes a lot of time and it is not available in most of the centers. In that case, what you can do, you can have a rapid detection of the HSV DNA in CSF. How will you detect the DNA in CSF? By polymerase chain reaction, PCR. So that is the best test for HSV rapid diagnosis. That is the most sensitive and specific test. So the others you can write for any virus, any kind of viral lab diagnosis. You can write the specimens, what are the direct demonstration available, if light microscopy is available, like in the case of production of inclusion bodies. Then you can write about the cell culture. You can write about serology, and molecular test is always a very good diagnostic test for viral infections. How will you treat herpes virus infection, herpes simplex virus? You have antiviral therapy available here, specific antiviral therapy. So that is acyclovir, valacyclovir, ganciclovir. So this group of drugs can be given for the herpes family. It's very effective. 400 milligram is the dose, and you may have to give it for approximately 5 times a day. So you have to repeat it for you have to give it for 5 doses per day. That is required for eradication of the herpes virus infection. So that is about herpes simplex virus. So what is the next category or the next virus that comes in this alpha herpes virinae subfamily? It is the varicella zoster virus. What is the human herpes virus number for varicella zoster? It is HHV3. It belongs to alpha herpes virinae subfamily. Okay, and this causes what? Varicella zoster causes two conditions. It causes varicella and also herpes zoster. Varicella is the chicken pox, and herpes zoster is actually a different manifestation of chicken pox, which is due to reactivation of the latent virus when the patient's immunity falls. So chickenpox is the primary. Chickenpox is what is referred to as varicella. It follows primary infection in a non-immune. Usually, most of us in childhood, we have acquired chickenpox. We are all non-immune to this virus. So you get it for the first time, you have chickenpox. But the same person, this virus will remain latent in the nerve ganglia because this is a herpes virus. At a later point, when the patient's immunity falls, the virus can get reactivated. It will go back to the distribution of the nerve and produce localized vesicular lesions in that nerve distribution, and that is called as herpes zoster. So zoster is actually a reactivation disease of herpes virus because of the latency. The mode of transmission of chickenpox, it is through respiratory route. It's like droplets and airborne transmission for chickenpox. And source of infection can be a case of chickenpox or herpes zoster. Both primary and reactivation disease can act as a source of infection. And they are highly infectious disease. Once if there is a case of chickenpox in a house, most of contacts, primary and secondary contacts, have the risk of getting the infection. So what is a classical clinical feature of chickenpox? They will be having vesicular rash. We classically describe the rash as dewdrop like. It will be looking like a dewdrop. So vesicular rash, mostly it is seen on the trunk of, that is mostly the first rashes appear over the trunk. Later it will spread to the other regions, but palm and sole will be spared. Face will be involved, mucosa may be involved, but palm and soles are spared. And rash will be having different stages. It starts as a macule, it goes to papule stage, it becomes vesicle, and finally it becomes a pustule. The vesicle may get infected, becoming pustule, and the scab will fall off. So in chickenpox, the rash appears in crops. That means you will get a few number of rashes appears together on day one. On the next day, there will be another set of rashes which are appearing. Like that, it progresses. So the first day set of rashes will be in the papule stage on the second day, but the second day set of rash will be in the macule stage. So at a time when you examine the patient, you can see all the stages of rashes over the body: macule stage, papule stage, vesicle stage, pustule stage, and scabs. So all the lesions have to scab off finally. That is what the sequelae of the rash is, and it may leave some scar. So you have to know what are the oral manifestations of chickenpox. It can involve mucosa, I told. So they can produce small blister-like lesions. Basically, they are also vesicles, fluid-filled vesicles. It can involve the buccal mucosa, gingiva, tongue, palate, and pharyngeal mucosa. They will be like, the mucosal lesions will be like a raised vesicle with surrounding erythema. They may rupture to form small ulcers with a red margin. That is what we see in the oral mucosal lesions of chickenpox. Now, coming to herpes zoster, if chickenpox is a childhood disease, a primary disease of childhood, zoster is actually an old age disease or an adult disease. It occurs in person who had chickenpox in the past, and the virus remains latent in the sensory ganglia, which later on leads to reactivation, leading to inflammation of the nerve. So this will lead to inflammation of the nerve. Zoster, it will lead to severe neuritic pain. So you will see a cluster of vesicles along the distribution of that nerve, the area where the nerve supplies, and these vesicles will be painful and there will be severe neuritic pain. This is because of inflammation of the nerve associated with this. Usually the vesicles are confined to the area supplied by a single sensory ganglion. So wherever the virus is remaining latent, so what is the area that is supplied by this sensory ganglion? Only in that area you will see the group of vesicles, and usually it is found to be unilateral. Now in zoster, the oral manifestations can be like, it can involve skin areas supplied by the various nerves, ophthalmic, mandibular, or maxillary nerve, usually unilateral involvement. And the oral lesions, the difference from varicella zoster lesions is they are extremely painful vesicles, which are seen on the buccal mucosa, over the tongue, pharynx, and larynx. So you can see extremely painful vesicles over the mucosa of mouth, oral cavity, tongue, pharynx, and larynx. So we'll see the difference between varicella zoster causing chickenpox and herpes zoster. Both of them are caused by the same virus, we know that. And what is the period of communicability? This is very important. It is actually starting a day before the eruption of the first rash, and it will be till 5 days after the last crust. So until the last crust falls off, and 5 days after that, the patient is infected. So 1 day before to 5 days after the last crust is the period of communicability of chickenpox. It is almost the same in herpes zoster. The evolution of rashes, I told you, it has all the stages like macule, papule, vesicle, pustule, and scab. Zoster also have a similar kind of evolution. Distribution of rash is what is the difference in zoster and chickenpox. In chickenpox, it appears first on the trunk and then it becomes generalized. Whereas in zoster, it is always localized, it is clustered, it is unilateral, and there will be severe neuritic pain. That is the difference in zoster and chickenpox. And I'll tell you one more name for herpes zoster. Zoster is commonly called as shingles. If suppose a question is like this, you have to know this is nothing but herpes zoster. So the localized distribution of the vesicles, unilateral involvement, neuritic pain, it is what that makes zoster. That is how you can identify zoster from chickenpox. Otherwise, most of the things are similar. Now, you know about the immunity of chickenpox. We say once you have a chickenpox infection, we have lifelong immunity, but the antibody formed fails to eliminate the virus from the sensory ganglia. So that is why the people, zoster, herpes zoster occur in people who are immune to varicella. This is because they are not acquiring the infection again. It is the infection which is already persistent in the nervous ganglia, in the neuron ganglia. And how will you diagnose chickenpox? Chickenpox is mostly actually a clinical diagnosis. We don't have to depend on the laboratory diagnosis in most of the cases, but at times if you need confirmation, what you can do is you can do a sang smear like what you have done in herpes. We take the scrapings from the base of the vesicle and you stain it with toluidine blue. You will be able to see the multinucleated giant cells. And also you can see the type A intranuclear inclusion bodies in this case also. So toluidine blue or GIMSA stain is commonly used. We have other tests like culture of virus in human fibroblast, HeLa or Vero. Mostly it is similar to herpes, no? Whatever we have seen in herpes, the same thing is applicable here. You can also detect specific IgM antibody in patient serum using ELISA, or you can go for a PCR test also. But most of the time the clinical picture is very clear. You don't have to depend on a laboratory diagnosis. And treatment is again acyclovir, that what we give for chickenpox. Acyclovir 4 times a day is given for treatment of varicella zoster virus. So lesions caused by HSV, or it can be simply asked as HSV. You can get a question as varicella zoster virus, or you can get the question as herpes zoster. Anything can be asked from this. Similarly, sang smear can also be asked. And classification of herpes is yet another question that can be asked from this area, classification of herpes viridae. So all these are the probable questions from this chapter of herpes, that is we have just covered herpes simplex virus and varicella zoster virus. You have a few more herpes viruses which will be dealt in a separate session. Thank you.